All right, hi, my name is Jennifer Shi, and I'm a postdoc in Marganka Sir's lab at MIT. And in this presentation, I'd like to share some preliminary data on a project that I've been doing, which assesses the computational power of calcium events in astrocytes of the mouse visual cortex. And so the main overarching question that we want to address is how astrocyte calcium events encode neuronal activity and behavior. And so astrocytes are the primary non-neuronal cell type in the brain. And we've now just come to appreciate their role not just as support cells to neurons, but also as having a more active role in neurotransmission and synaptic function. Astrocytes are not electrically excitable and don't spike like neurons do. Instead, their activity is read out in calcium signals in various subcellular compartments, from their soma to their primary branches to fine processes. And so you can see that here in this video, where there's a red static marker that marks out the soma, the primary branches, and then in the and extending to the edges of the territory, they're fine processes. And so much of the recent work in the astrocyte field has focused on how these signals not only correlate with neuronal activity, but also may influence it. And so in this project, I'm coming at this idea by looking at how astrocyte calcium events, particularly in the microdomains of the fine processes, which you can see again as these dynamic green signals in the video, encode neuronal activity on a network level and the subsequent behavioral output. And so given the diverse events in astrocytes in microdomains, which you can appreciate by the diverse signals in the video, and the fact that they encompass several neurons within their territory, the aim is to translate this astrocyte microdomain event code to understand how they represent activity across several neurons and downstream behavioral output. To answer this question, we performed dual calcium imaging experiments using transgenic astrocytic GCAM mice combined with viral expression of the neuronal JR gecko. The mouse is presenting with drifting grading visual stimulus, and during the imaging session, we also record pupil diameter as a readout of brain state and arousal, as well as locomotion. And so my analysis currently focuses on using neuronal spiking activity combined with these behavioral parameters that I just described as predictors for generalized linear models, which we then use to predict microdomain events in astrocytes. And so here you can see a video example of neuronal activity in dendrites during visual stimulus and behavior. And so you're looking at the red channel, which is showing dendritic activity. And our goal again is to understand how the microdomain events in astrocytes, which are these green signals that you'll see occasionally popping up, encode these various parameters here on the right. So we use an event-based detection method to extract the astrocyte calcium signals from each imaging session. Given that these events are by definition spatially temporally unique, to be able to model and predict astrocyte activity throughout an imaging and visual stimulus session, we cluster all of the events that we detected using these 5 parameters, and then used an unsupervised k-means method to group them into these 5 clusters. Here on the right, the heat map shows all of the events from one cluster per line. So that's going across throughout the duration of the imaging session. And basically the signals here are where they happen in time. So essentially this is a peristimulus time histogram of each astrocyte cluster. And then we've built the GLM for each one of these groups with the idea that the results from our modeling will then give us an idea of what to expect, will give us an idea of what predictors are contributing to the event activity in each functionally defined group. And so here are some of the results from the analysis, which shows mainly that neuronal activity contributes most to astrocyte microdomain activity, as shown by the higher coefficients of these predictors in each of these models. We also find differences in how the predictors contribute to the GLM for each cluster, such as more positive neuronal weights for cluster 5, and more negative weights of running activity for cluster 2, which is here. So at the end of the day, GLMs still provide only correlative information, but these results suggest important relationships between these predictors and the resulting astrocyte activity. So for instance, cluster 5 is characterized largely by events that are temporally correlated. And so you can see that in the top row of this k-means clustering heat map here, which means that they happen at the same time in a coordinated fashion. And neuronal activity seems to be positively correlated with this type of astrocyte activity. On the other hand, cluster 2 is characterized by events that don't have any of these particular features. And you can observe that here in this group of events. And is negatively correlated with locomotion, suggesting that these features which were used to cluster the events are critical to how astrocytes encode this particular type of behavior. And so to summarize, GLMs were used in this study to model functional clusters of astrocyte microdomain during visual stimulus presentation. Neuronal predictors contribute most to the models, and differences in the predictor contribution to individual clusters can guide us in our future experiments and analyses of encoding by astrocyte microdomain activity.